do, now I understand why you're saying where Mabel is, because she's listed on there. Okay, so I'm just going to share my screen again and we'll get started.
All right, guys, this is being recorded as well, just to keep you mindful of that, so that we'll hopefully be able to play it back at some stage later on for you once it's all complete.
So you can use it for revision and you can jump back into Canvas and do your post work again, try and get as much out of this program as you can. Okay, so quickly down the bottom, you'll see that there is a little face, then there is a microphone. This is on our Collaborate. Then there is your microphone, followed by that is your camera, and the last one is your hands up. So what I'd like you to do is raise your hand just so we know that you know how to use this. Most of you will be all over, this will be sweet. Awesome. And once you've raised it, just lower it just so we know that you know how to do that, because that makes it easier for us too. Awesome. Okay, the last one I want to show you, if any of you are having any problems at any stage, at the very bottom you'll see a purple little arrow in the corner. Click on there and you'll see a little first one or tab come out, and the first one is your chat. And what we'd like you to do is just write hi or put an emoji in there of how you're feeling right now in the everyone chat, just so you know where it is if you have any issues later on. Perfect. Okay, ET is going to put up a question in your chat for you in just a moment. Don't worry about that. How do these images that you see in front of you, how do they link together? So the first one is your bacteria, the next one is a frog that is glowing, and the last one is insulin. Bit of a hint there, have a look at your bacteria. There is a funny round piece of DNA floating in it called a plasmid. So how are these images all linked? We've got an answer there. Oh, ET. So think about what you did for your pre-work and what our program's about today. So you've got your bacteria, you've got that little tiny bit of DNA in a ring or a circular piece of DNA, a frog that's glowing, and your insulin. Any guesses, anyone? So just typing in the chat or putting on your mic? Or is everyone falling asleep after lunch? No, I'm getting crickets today. That's all right, guys. So by the end of today's session, hopefully we can answer this, but the biggest thing is you've got your bacteria here. That bacteria, of course, is what we put plasmids into to clone our bit of DNA. Thank you. So you've got your DNA there. You've got your frog that is glowing because it's got that particular gene inserted into it. And then, of course, your insulin, which is one of our genes that we require in order to, or from our pancreas, to create insulin to combat our sugar levels. So we're going to explain what this all means in one moment as we head through. So we've already done a bit of our navigating online today. We're going to have a look at our story of insulin. Then we're going to be doing our bacterial transformation, where you guys will be doing it beside us. So I'll show you in the lab what to do, and I've also got Etienne who's going to jump in and show you how to streak plates, because he's a pro, I'm not. And then you guys are going to be doing this in the virtual lab as well. We're going to explain how our particular frog you just saw is actually glowing, and this is due to the GFP gene. Then we're going to do predictions of our plates. So we're going to think about why are our plates actually having different growth. Then we're going to be streaking them. That's when I said Etienne's going to be our star and come in and show you how to do that. And then you guys are going to do it in your virtual lab. Then we're going to do our observations of our plates, and we've got our plates all here ready to show you. And we also have your plates that you'll be looking at in your virtual lab. Then the last stage is our plasma digest, and we're going to be running that in a gel, and we already have a gel that's been run with some pretty good results. We're pretty happy with them. And we're going to show you how to set it up and then show you the results. Due to time constrictions, we can't do everything. But real world, you guys would be in here in the lab doing this whole prac. So sadly, we can't because of COVID, but hopefully you guys get something out of this today. And the last thing, of course, is a conclusion, because we always want to tie all our loose ends together. So just a bit of housekeeping. We're going to end up with only 3 tabs open today. The only 2 that we want open constantly is our Canvas LMS and our Collaborate. Collaborate, you are already in. Canvas, can you please log in if you have not done so? And when you are logged in, just raise your hand, just so we know that you're ready to answer any questions in there. So just all raise your hand when we have both Collaborate and Canvas. Thanks so much, guys. If anyone's having any issues with logging on, just type in the chat and Etienne can help you out. And if you're unsure what your login is or anything, Etienne's just going to throw up the KIOSCinfrastructure.com website if you don't have it. So we got everyone. So we're still waiting on a few people. So once you're logged into Canvas and Collaborate, just raise your hand.
That's me, sorry.
All right, Pete. So you'll see Etienne's just put on the website if you need, if you don't know where you're going. And remember, your teacher has already given you your log on, which of course is your Wantirna email, and then your password, which was sent to you. If anyone's having any issues, please just email, or sorry, not email, chat to Etienne and he'll be able to help you out.
Remember, kiosk with a C, not a K.
Yep. And remember to leave your hand up if you're in both systems. So we keep it still together. So Andrew, Grace, Jacqueline, Jacob, Justin, Mackenzie, Madison, Nathan, and Taya. Waiting on you guys.
Remember, if you're having any issues, please type in the chat. Thanks, Andrew. We'll get Etienne to help you out. Okay, Grace, Jocelyn, Jacob, is it Justin? And Madison.
Okay, Maddie's up.
Awesome. Remember to raise your hand once you're in.
Grace, Jacqueline, Joshin, Jacob. Joshin, are you there, mate? All right, we might need to just push on because they've gone to get themselves some lunch.
No worries. So the other tab we'll be opening later and we'll put the link up for you in just a moment. So everyone just lower your hands. Anyone that's having any issues, just type to Etienne and he'll help you out. So here are our learning intentions for today. So the first thing is we want to determine and analyse the results of a restriction digest, so a restriction enzyme, and we're going to be using EcoR1. And the really cool thing is when you're actually doing this practice, you can do this at the start or you can do it at the end. So there's lots of different ways you can do it. The next thing we're going to be doing is observing and you guys will also be doing it virtually, a lab techniques related to bacterial transformation. The last thing is we're going to observe and evaluate those particular growths to actually determine which bacteria have transformed. So firstly, we're just going to look at a short little video on the story of insulin. Is there any sound? Okay, hang on just one moment, guys. We'll get your sound. For some reason, it's not doing it. Yeah. Bear with me one moment, guys. Makes it a bit easier having sound on.
Now introduced to the world. In 1922, at the University of Toronto, the scientists went to a hospital ward full of diabetic children, most of them comatose and dying from diabetic ketoacidosis. This is known as one of medicine's most incredible moments. Imagine a room full of parents sitting at the bedside, waiting for the inevitable death of their child. The scientists went from bed to bed and injected the children with the new purified extract, insulin. As they began to inject the last comatose child, the first child injected began to awaken. Then, one by one, all the children awoke from their diabetic comas. A room of death and gloom became a place of joy and hope. Thanks, Dr. Banting and Dr. Best.
So insulin, it's a pretty interesting story. It wasn't until 1922 when they actually had something to give someone who had diabetes, which is pretty crazy to think. Previous to that, people would just die. And then it wasn't until 1982 when they were able to actually give them a different form of insulin. And this came from a little bacteria with a plasmid growing it, which is amazing to think, because previous to this, they had a lot of issues with people becoming allergic to the insulin that came from your pig's or your cow's pancreas. And also there's issues with religion for certain people as well. So let's get into our first prac. The first thing we're going to be doing is transforming our bacteria and determining the results through our bacterial growth. And all your materials and methods are on Canvas, so you don't need to worry about it. Now, as I said before to Mr. Cos, there is a Pipette Skills little virtual lab that you guys can do if you want to get some experience, but I'm going to be showing you how to use it in the lab here. So I'm going to stop sharing my screen now, and we're going to start getting ready to do our lab. Okay, so we've got everything here we need. Okay, so we have our micropipettes here at the top. Raise your hand if anyone's used a micropipette before, just so we see how many experts we have in the room today. So a few of you have, that's all good. If you've come to do forensic science, you probably would have used them 2 years ago if you came to KIOSC, or if you did the year 10 genetics and microarray program, you would have used it as well. Now with our micropipettes, we need our micropipette tips. We also need our waste. We also need, in this area, we're going to be using our Bunsen burner. So remember back to year 7 when you got your Bunsen burner license. We have our inoculation loop, and at the end there it's a tiny little loop. It is made from nichrome wire. That way we can heat it in our Bunsen burner and kill anything that's on there. We also have our P-glow and our transformation liquid here. And we have 2 of our Effendorf tubes, which are empty, which we're going to be loading up into. So this liquid that I have here has just come off the ice because we want to do a thing called heat shock. So we're going from cold, we then put it into here, get it all ready to go, and then put it into our water bath where we're going to heat it up to 42 degrees to basically shock it. And you've got a salt here being your transformation solution. So it's going to allow your membrane of your bacteria to open up, allowing your plasmids in. And you're going to need a control, and you're also going to need one that actually has the plasmid in it. So the first thing I'm going to be doing is writing on them which one is which. Now, in our case, our plasmid is the P-glow plasmid. So we're going to be putting our plus P-glow and our minus P-glow on top. So if I'm putting a plus on it, it means that I'm adding the plasmid into it. If I'm putting a minus on it, it simply means that there is no plasmid in there. So we'll just have bacteria and transformation liquid in there. So now it's all labelled. We now will be getting our micropipette. And I want one that does 16. Okay, so here's your micropipette. A few things to remember. At the top here, you've got your button. Now, in order to use your micropipette, you need to find two things. One is your first stop, which is where you just squeeze it halfway down. And that is basically pushing all that air out of the barrel of your micropipette. So when you go to use it and you've got your tip on it, you simply push it down to that first stop, put it into a container, draw up the liquid by letting that go back up, the plunger, and then you go to the container that you want to put it in, and then you go to what's called our second stop, which is all the way down at the bottom. So you would put it into the next container, you would then push it all the way down to the bottom, and that then would pull everything out, and then you would pull out your micropipette and release. If you release when your micropipette's inside the container, you'll actually start to draw up the liquid as well, which could be interesting. But no, I need to have this one here first because I'm doing my transformation liquid. The other thing is you'll notice with our tip boxes, they always have the lid closed because you don't want to open it up and leave it open because anyway, the problem is contaminants and things will get on there. So you open it up. And I'm going to get my pipette set to 250 microliters. You can see at the moment it's at 260. So simply turning the top here, it will take you down to your 250. So you're going to open up your transformation liquid, go down to that first stop, go into the solution, and you draw it up. And then I'm going to go into my minus P-glow, and then you're just going to pipette it all the way into there. And then to eject, you just push that button there, and it ejects it off. ET, can I just grab just a little bit more because from the fridge? Thank you. Sorry guys, we're just missing a little bit of it. All right, so I'm going to be doing exactly the same for our, thanks so much for getting it from the fridge. So I'm going to be doing this again for our P-glow plus. So simply, first stop, jump it in there. Oh, that one's gone a bit funny. There we go, that's a bit better. And then we put it into our P-glow plus. Nicely done. And now we're going to grab our bacteria that we've had growing on our plate. Now this is just E. coli. It doesn't have anything else in with it at all. So here we are. Where do you want it?
Yeah, I'll
come to you down there because it's black. It's a bit easier to see. Okay, and you can see here how it's been streaked. So you've got the first bit here where it's run, and then you've got 3 lines this way, 3 to 4 lines that way, and then 3 to 4 lines that way. What we're going to be trying to get with our inoculation loop is just 1 to 2 colonies. So 1 to 2 colonies are like these little dots here. It's a bit hard to see. You don't want to be taking a whole heap of the bacteria, otherwise you're having too much.
Come in nice.
So we'll come in a bit closer to show you. So we just only want to show just the little circles.
All right, guys, so I'm going to jump in here quickly because we did a prac where we had a lawn culture of E. coli, so it was spread over the entire plate. Whereas this one here, it's really heavily concentrated on the left-hand side there, And then the way that they've plated it out is to minimise that concentration, so we can get individual colonies. So those individual colonies started off from one bacteria that replicated and grew and grew and grew.
Thanks for that, Pete. So yeah, so once you've got your colonies, you then want to spread out, because we only want to get one little colony, or you don't want to have a lawn that you're picking from. So first thing I'm going to do is light my Bunsen burner. There we go. Okay, let's move this stuff out of the way. Now the other thing I forgot to do before I get, oh no, I can do it afterwards, it's fine. What we're going to do is put it to our working flame, and then we're going to heat it up. Now notice how it's starting to get very red hot. So that's just burning anything off our nichrome wire. So you can see that loop starting to get really hot. Yes, I'm just showing them. I'm not going to put it onto my bacteria that hot, Ian. So you've got to make sure once you've done it that you don't just go straight into your bacteria, because what is happening with your bacteria when it gets too hot? It will go kaput. So the other thing is, when you do this, you can put it onto the center, and if it's too hot, because you've got no bacteria growing there, you can actually cool it off as well. So I'm just going to grab 1 to 2 little colonies. Okay. Put that back down. And we stay near our flame, because this is what is keeping this all aseptic techniques all nice and safe, and so we're not getting any other things in there. And we'll do the same for our P-Glow minus. So cool it down again. Grab just 1 to 2 colonies. There we go. So we go into our minus. And the last step I need to do, now you can do this 2 ways, as I found out the other day, is you can either use your loop, or you can pipette in the P-Glow minus. So now I've heated that up. I'm now going to, not my P-Glow, my P-Glow plasmid, sorry. So now I'm going to warm that up, put it into here, and just turn it around a little bit. Then we're going to put it into our P-Glow plus, because we want to have our plasmid in with our bacteria that is labelled that way. And then once again, you heat it up, kill any of those bacteria or plasmids that are on there. Then my next step I need to do, because we've gone from cold, we are then going to put this into our water bath. And when we put it into our water bath, it's at 42, sorry, for about 25 minutes. Is that right? No, 50 seconds. I'm doing the wrong one again. For 50 seconds, we will be putting it in here. And it simply just floats in there for 50 seconds. ET's all over it. He's got his little timer for me. And he'll be pulling that out for us. So now what we're going to do is jump back in to talking about our plates. And I'm just going to put that back onto safety flame. So as we said, if you were in the lab, you guys would be doing all this. Okay, so sounds like you guys are pros already. Is that cut off as well? Yeah. That's weird, it's doing weird things. Okay, so here's our terminology. Which one of these plates in the chat, can you just write A, B or C is the lawn growth? So A, B or C, which one is our lawn growth here? Nice work. Yep, you're all over it. So therefore, B, what do we call B? Thanks, CT. Can anyone remember what I was just calling everything that I was just picking up with my loop? I was looking for 1 to 2. Well done. Awesome work, guys. You are listening. Therefore, the last one doesn't have any growth, so we call that our no growth. So now we're just going to explain to you a bit more about our gene here. So we've got our lovely plasmid here, and you've got your promoter, you've got your GFP, and this is your green fluorescent protein. This is basically a marker. When it has arabinose sugar present, which is your promoter, it then makes this protein come out. When it's got UV light around, it will then fluoresce in this green light. Now this part here, the last part, is your gene of interest. So this is basically, we use GFP, so your green fluorescent protein, as a marker to say, yes, this particular bacteria has taken up the plasmid, therefore it's glowing. And the really cool thing is this little bit of bacterial protein actually comes from sea jellies. So a pretty cool little thing that it does. The next thing we're going to be getting you guys to do, and I'm going to show you two different things, we're going to go into Canvas and do our predictions. So remember with our plates, and I'm going to show you in Canvas now. So I'm just going to flick my share. Bear one moment. This is why we wanted you guys to have Canvas all ready to go. And I want that one. Okay, so we're just going to jump into, doesn't matter which one I pick, it's all the same. Okay, so we're going to jump into module 4. No, module 3, what am I doing? Module 3.4, and you're going to be doing your predictions. So I'm just going to show you what you need to do first. So your first plate, which we'll be plating up, is our control. What does control mean to you guys when we say control in an experiment? So whenever you do an experiment, you need to have a control. Here we go. Oh, I love that answer. Not exposed to the independent variable. Well done. That is awesome. So yeah, so we're not changing anything. We want to keep everything constant. So in this case, you're going to have your lovely agar just sitting in the bottom. You are also just going to have your bacteria growing on it. So the first plate, you have nothing apart from your LB, which is your broth, which allows your bacteria to grow on. And the difference is, you'll see that it's called plus AMP. This is because in the virtual lab, which is what we wanted you to get your predictions on, they are using whether or not it has ampicillin or no ampicillin there. So in our case, we want it to grow. Therefore, if it takes up the plasmid and there's ampicillin present, it will grow. If it doesn't have the ampicillin present, it won't grow because it doesn't have that plasmid. So jumping down here, the next plate is where we have our ampicillin present, so it's not a control. And you do have your plus AMP. The next one is our control, and it does not have ampicillin on this plate, and you've got no plasmid uptake. The last one, you're on a plate with ampicillin, and it doesn't have your ampicillin uptake. So for us, we're calling this our minus P glow, whereas on your virtual lab, which you'll be jumping to in a moment, they call it minus amp R. So what I'd like you to do is jump into Canvas and go to 3.4, your plate predictions. I'm going to give you 1 minute, and when you get back, there'll be a question waiting for you in the poll. So 1 minute, guys.
Now, guys, these are predictions. You don't have to stress about your accuracy of the prediction. Remember, these are predictions, and we'll hopefully get some tying down what actually occurs later on once we see our results.
So as Mr. Cos has said, it is only a prediction. Don't freak out. Remember, this is the time where we make mistakes, not in your exam or SAC. So your first poll is up already. So we're predicting, what did you guys think will happen on plate number 1? Now, just a bit of a heads up, we call a lawn growth when we're streaking what you saw before. So remember on that first plate, it was a control, and it just had LB, which is your broth, growing or in your agar. So if you haven't done so already, jump back into Collaborate and answer the poll waiting for you. Okay, just waiting on 4 more, or is it 3 more? All right, so we can see we've got a combination of lawn and glowing colonies are your top 2. All right, so we're going to chuck up the next plate. What do you think is going to be in your second plate? Remember with this, you are looking at a plate that has your ampicillin on it. Remember what is in your little bacteria. Does it have a plasmid or no plasmid in that second plate? Benjamin, you right there? Do you have a question? Because your mic's on.
It's been on since the start.
That's interesting. Yeah. All good, no worries. Thanks, Ben. Has a mind of its own. Oh, interesting. All right, so have a look there, guys. If you haven't answered, please do so. That's all right. Okay, so you can see some of you are saying lawn growth, some of you are saying your glowing colonies. All right, so 2 more plates to go. We'll just quickly go through it. That's all good. So remember on this plate, it's another control plate. Figure out whether or not it has the plasmid or not. See if that changes your ideas. Okay, showing you the results. So, oh, a bit of a tie there. Some people haven't responded. That's cool. All right, and the last plate. So it's always interesting to see how you guys are predicting, to think about what is growing on that plate, as into what was on the plate in the very beginning. So remember plate 4, we had our ampicillin present, but can you remember if it had the plasmid or not? Okay, showing the response there. So you can see you guys are getting some, oh, it's a nice bell curve in a way. Some interesting responses there. Okay, so the next thing I would like to show you is you guys are going to jump into your WOW Biolab, and I'm just going to show you a few things first. Now the thing is, when you jump in here, you need to make sure you allow your Flash player to work. If you're having issues, just let us know. What you're going to do is jump into bacterial transformation. Also, just a heads up, we do have a gel electrophoresis here where you guys can do at a later date in your last module. It goes through here, tells you about insulin. Then from here, you need to go in here and click everything. If you don't click everything and it gets ticked off, it won't let you go on. So I'll just quickly go around and tick them all. Doesn't take long at all. So I just need my pipette and now E. coli down here. Okay, then you're going to click on to your procedure, and this gives you step by step. Just like your predictions you did, you're just going to do again. So you can just type lawn, colonies, etc. Then once you've done that, you need to go through and get up to step 4. So once you're at step 4, jump back into Collaborate, and we're going to show you how to plate up and strip your own plate. So do not go up to step 5. You have 5 minutes to jump in there and get up to step 4. Etienne's going to be putting up the link for you in the chat. So if you can't find it, it's there for you. It's also on your canvas as well. So 5 minutes, guys, and then jumping back into Collaborate.
Hi guys, just a 2-minute call. If you could get through that as quickly as possible and make it back in the next. So yeah, if you can go up to step 4, and then once you've made it back, if you can just put your hands up for me, that would be excellent. Thank you. Okay, guys, 1 minute. If you can just finish up what you're doing and come back, we'll move on with the next section. Alrighty, guys, if we can have you back, don't stress if you haven't managed to get that done. We're just trying to get you into the zone, and you can complete that at a later date, so don't stress too much. So could I get your hand up as well if you're back? Oh, we're getting there.
30 back.
Okay, so we're waiting the last, what, 10 maybe? Last 12.
Just remind them again.
So if you could just raise your hand if you're back, just so we can move on, that'd be excellent. Thank you.
We've got 35, so let's push on. Lower hands.
Okay. So hi guys, how you doing? My name's ET, that's what I get called here. I'm the guy who does all the hard work and doesn't get enough pay or credit. So can I get some F's in the chat if everyone heard that? So what we're going to be doing is we're going to be swabbing a plate. So we're just going to crank up the heat just to create that sterile environment, just create a bit of a mushroom, so anything that falls into this area kind of flies back up and away from our sample. So we're going to get our swab here. We're just going to dip this into our plus P-glow sample. So this is the bacteria that's been heat shocked and has taken up the plasmid. Just going to do a bit of a twist there. So we don't need to see any bacteria because we obviously know there's millions and millions in there. Now this is the pattern we're going to be following. So as you can see at the top, we have our primary streak, as your teacher explained before. And then each time we go down, what we do is we get less and less bacteria. So anyone in the chat tell me what this process is called? It has a special name. It starts with the letter D. So we're just going to paint it here. Just want to cover the surface pretty much. After we've done that, we can deposit our swab into the waste. Now, we got any answers there, Alex? Or
Not yet.
not yet? That's fine. If anyone wants to take a guess, the process starts with D, and it's basically when we go from a lower concentration, sorry, high concentration to a lower concentration. Okay, so now that we've got our primary, what's the word, primary streak, we're going to dip into this. So we've heated and disinfected our loop. Just going to follow these lines, these parallel lines here, dip into the primary, and now we have 3 different streaks. So this process, if anyone managed to come up with the answer, was dilution. Okay, so each time we go through, the bacteria gets more and more diluted. So instead of having the millions and millions of bacteria that we originally had, by the time we get to the end, theoretically, we should have as little as hopefully only a couple, but that can go down to, if we can get within the 50 to 100 range, we're doing pretty well. So we go through one more time. Of course, heat it up and cook our bacteria off. We want them medium rare, excellent. And then we're going to do our last 3. Excellent. So that basically cooks off and disinfects everything. My coworkers like to think I'm hilarious, which is excellent. So now that's been done, we return to our safety flame, and we now have our streaked plate. So what we do with this, given we had the extra time, we'll put this in the incubator and see if this would grow. Yeah, so that's pretty much it. So I'll hand you back off to Kat, and this concludes my TED talk. Thank you.
Oh, you did get dilution. Nice work, guys. Okay, so now we've done that. Let me just see what we're up to next. We are up to you guys streaking the plates. So what we'd like you to do is jump back into your virtual lab. We're going to give you another 5 minutes to do this in. So this is up to step 9. Now, just remember with your plates, you've got lots of different things in there. So take note as to which ones are being put onto which one. Sorry guys, I'm just doing a promo photo right now, just one moment.
Hurry up, man.
Leanne, how are you doing it? I'm done. Yep, put the poll up now. Hi guys, so 2 more minutes, you'll see a poll waiting for you when you get back with your predictions of your results. And if you're not finished in 2 minutes time, don't panic, you have time to go back and do your virtual lab later. All right guys, 1 minute call and then jumping back onto Collaborate and answering the poll question for us. All right guys, jumping back onto Collaborate and answering your poll. And then we're going to be showing you our results. And we're going to see if you have any differences. So how many are we waiting on?
We are waiting on about 5.
Okay.
4, 3, 2.
All right, showing the results. So that's all right that our predictions are not perfect, because in the real world, when it comes to science, it doesn't always go that way. Okay, so we're going to jump over now and have a look at our results here. So we're going to turn off the lights and it will go a bit dark for a little bit. Can we just make sure the poll is being removed as well? Cool. Yep, thank you. So we're just going to have to set up so we can show you. And remember, when we talk about lawn growth, we are talking about in regards to our streaking. So it's a little bit different. So can we see it? Let me have a look. I'm going to turn that a little bit so you guys, we can see what you are seeing.
Yeah, my bad.
No, you're all good. Actually, it's probably easier if I just do this. That makes more sense.
Where you want to start?
Okay, so we're going to start at this first plate here. So this one, remember, is our control, and it has our plus P glow plasmid in it. And you can see that we expect it to have growth. So you can see it had a lot here, and as you've moved out, you slowly get to your little colonies, so it's been diluted. There is another technique where you can actually use a sterile pipette where you just squirt it into the centre and spread it out, and that will give you a lawn as well. The second one is over here, plate number 2. Now, the interesting thing is we have an additional one. Remember, we're talking about requiring our sugar to make it glow. So as you see here, you've got your LB, which is your broth. You have your ampicillin. So your ampicillin will stop things growing if it does not have the plasmid. And then you've got your arabinose. And the arabinose is our sugar that will make the protein being produced, which will allow it to glow under UV light. Then we have our other 2 down the bottom here. We have plate 3, and this is our minus P glow, and this just has LB on it. So we expect it to have growth because there's nothing stopping it here. And because it's got minus P glow, it does not have the plasmid in it. So you can see you've got your nice big chunk here, and as you slowly go on out, you've got your dilution happening. The fourth plate is our P glow minus as well, but in this case, we have ampicillin. And because ampicillin's on here and it hasn't taken up the plasmid, we expect no growth. Now I'm going to show you, we've got some additional plates to explain some other things as well. I'm just going to chuck this one under here. So this here is one of our older plates. And you can see, it's very hard to see, but there is colonies sitting on here. This one does not have the, what am I trying to say? The arabinose, thank you, I've gone blank again. Therefore it won't glow. Because this plate here has arabinose, you do get the glowing. And the other one I wanted to show you, yeah, it's a bit hard to see the glow sometimes.
Little spots.
So they're tiny little spots there.
Oh, out of focus.
Here we go, guys. Just getting back into focus, our apologies.
Come on, camera. There we go.
There we go. So you can start to see those little dots there, and those ones are glowing compared to the other one. Okay. So we're going to turn the lights back on. Okay. And I'm going to share my PowerPoint again just one more time. And remember, guys, if you've got any questions at any time, just let us know. So in regards to step 10, that's where you write your observations in your notebook. And these questions are the same questions that you have in your quiz in your Canvas. And looking at the time, we're just not going to do that now. We'll get you guys to follow that in a moment. And this is where you can do all your analysis of that. So once again, I just wanted to recap and show you our pictures here. So this is what we're talking about with lawn growth when it isn't all streaked out. So you've got plate 1 and plate 3 that have the lawn growth. Plate 2 has that beautiful colonies that are glowing, and plate 4, which doesn't have your plasmid present, therefore it's not glowing. So let's start our second prac, and we're going to be doing a digestion of our plasmid and running it in a gel. So why do you think we go through the process of digesting our plasmid? What's the purpose of this? And remember I said we could do this at the start of our prac or at the end of our prac. Why do you think we go through of getting our little plasmid and cutting out with our enzyme, what's the purpose of it? Think about what's in your little plasmid. Bit of a hint, insulin. What do we got there? Yeah, we want to make sure we've got that inserted gene in there, number one. And the other reason is we want to try and amplify that one. So remember with PCR, you're going to be amplifying genes. This is the old PCR. So you've got to make sure you've got your different sizing of your bands, and you want to purify the smaller band because that is your target DNA. So we're going to jump in and get this all ready for you guys to go. So we're going to go to our big screen. But there's just a few things I want to point out when we jump into it. So I just want to explain a few things. Okay, so we all know that our plasmid is just like a round rubber band in a way if we're looking at it, but it's tiny. And when we get our little restriction enzyme, we're going to have our scissors. If we make one cut, we're going to only end up with one band when it comes out in our gel. So we need our restriction enzyme to do 2 cuts, then that way we end up with 2 bands. Okay? The other thing is our little plasmid here. I might do it on here because it's nice and black. Yep. So you've got your plasmid here, you've got your promoter here, you've got your green fluorescent protein here, and then you've got your gene of interest. In our case, it is our insulin gene, and then you've got some of your plasma DNA that's left over here. So our EcoR enzyme is going to come in, it's going to eat here, which makes us have our sticky end. Okay, so you can see it there. Then, if it just cuts there, you're still only going to have one piece of DNA. Comes over here, cuts over here, and then that gives us our 2 pieces of DNA. Notice here with our E. coli R1 that is, that you get sticky ends. Can anyone tell me what another cut an enzyme will do? So our EcoR1 does sticky ends. Can you tell me what another restriction enzyme will cut? Well done, you guys are all over this. So there's your explanation. We're now going to jump in and actually do it for you. And due to time, we've actually already got these prepared. So you can see here, we've got our EcoR1 plus and EcoR1 negative. What we're putting in there is our DNA, which is our P-glow plasmid, our sterile water, and in our EcoR1 plus, we're putting in the enzyme. So once we do that, we then put from here, we put it into our floater again. And this time it goes into our water bath for 25 minutes. So you put it into our water bath, and then when we come out, we put our loading dye in it. So we're going to jump over around the other side. I'll take that and that. Yep. Because we've got our gel and everything already over here. Can I have a show of hands who has done gel electrophoresis before? So who's run a gel? So 4 of you? Okay. So the first thing I need to do, I'm going to grab that one. Awesome. So if you guys were here, you would have been having fun and doing it with us here, but sadly, due to COVID, we're not allowed to. Okay, so we're going to be putting our ladder in the first one. Go. And we're going to do it a bit differently. We're going to put our ladder in the center. And for you guys, it's really good to see having ladders in different spots. And I'll show you when you do your prediction, we have you doing it in the side. Okay, so we've got our ladder in there. Get a new tip, And we have our Echo R1 minus. So this one does not have the restriction enzyme. So we go into here. Okay, and the last one is our Echo R1 plus. So this one has our restriction enzyme. So go to that first stop, put it in, and I'll do this one nice and slow so you can zoom in. So you tell me when you're ready.
Might come right over. Yeah, nice and close. See every detail.
So you tell me when you're ready.
Yeah, I'm good.
Okay, so we go into the well and you inject it in. So you can see all three are in there. We already have our gel and buffer in there. Sure, I'll just get a tip, it'll be easier. Okay, so we have our first well here, or it's the second well. We're calling this one lane 2 because it's in lane 2. That is just our E. coli R1 negative. Yep. We'll just get you into focus. Sorry, guys.
Just trying to get it out of the. There we go. Yep. I think I can see some colour there. Yep, that's better.
Then we have our DNA ladder here, and then our Echo R1 plus, which one has the enzyme in it, in this one here. So the next thing we do is we turn on our power at the back here. Okay, we then set it to run on our program, and that will need to run for about 45 minutes. The nice thing in here is that we've got one that we've prepared earlier. So what we're going to do now is jump into Canvas, and I'm going to get you guys to do your predictions. I'm just going to explain one more thing. Okay, just conscious of the time, we don't want to keep you guys longer than you need to. So just to help you a bit with your predictions, just as I showed you here, your plasmid is circular. If you only do one cut, you're going to end up with it being linear. So Echo R1 cuts at 2 sites, so therefore this will help you with predicting how many particular bands you're going to see in your gel. So what I want you to do, I'm going to jump over to my other one, just bear with me one moment. That one there. Okay, and you're going to go down to module 4. And you're going to go into 4.5, your gel prediction. And what I wanted to show you here is we've actually got a different position in our photo. So your ladder is on the side here. So lane 2, this one is just Echo R minus, so it doesn't have the enzyme. Lane 3 is here, and this one does have the enzyme. So 1 minute, guys, jump into Canvas. When you're back, have your hand up so we know that you're back with us. So 1 minute to answer those questions for us. All right, guys, so when you get back, you'll see a poll question waiting for you. So just answer the poll question. All right, guys, so can you just jump back into Collaborate and quickly answer that poll there for you? That's on your prediction for your gel. So which one do you think in lane 2, how many it will have compared to lane 3? This also helps your teachers to see if you're listening. So if you got your hand raised, you can lower it now as well. Thank you. Thanks, guys. Okay, so we're going to show your responses now. So you can see most of you are, hopefully you're heading in the right direction. Okay. So as we were saying at the start, we also, can you just make the poll disappear? Thank you. As you can see, there's another really cool thing you can do. It's called Restriction Mapper, and you can do this in your own time. And basically, you go in there and you can put in the DNA that we're giving you for pGlow. Say it's a circular piece of DNA. You put in which restriction enzymes you want in there, and you can actually see how many cuts it makes and what DNA it spits out at the end. So when you saw me putting the DNA into our gel, you would have noticed that I was putting it at the largest end, as it says. That's where our wells are sitting, and that's our negative end. Why am I putting it there and not at the positive end? Your clue is, what charge does our DNA have? So what charge does our DNA have? So just type in the chat. Oh, look at you guys, you're all pros. Yes, it is negatively charged. So that's why it moves towards the positive end. If I was to put it down here at my positive end, it then would keep going the other way. So we've got to make sure it's in the right way around. The last thing is, size does matter when it comes to your gel electrophoresis. You actually want to be small to move faster. So the smaller you are, the faster you'll move through the gel. So the best way to think of it is an elephant and a little mouse. So the little mouse is going to get through that jungle a lot faster than that large elephant. So this is a hint to help you for working out sizes. So what I'd like you to do is type in the chat what you think the size, so the base pairs, so the number of base pairs, that's the fragment for E. What size do you think fragment E is at? So how many base pairs? Oh, you guys are on fire. Well done. 200 base pairs. So what we're going to have a look at now is our gel that we've got. So we just need to turn off the lights and get the camera ready. So bear with us for just one moment, and we're going to show you our results, and then we're going to show you a really good picture as well. You're right there, ET? Okay, we're just going to stop sharing, and I'm going to get ET to share the camera on his. So bear with us just one moment. We're just going to share our computer. So share camera. Awesome. Okay. If you can't see, just let us know. And we'll get rid of that. Okay. Here we go. So we're just getting into focus. If it's too blurry, just let us know. I'm going to get a little pointer for us.
How's that, guys? Can I get a hands up in the chat if that looks all right?
So just let us know if you can see it clearly. Awesome.
Awesome. Thanks, guys.
Okay, so at that center here, you can see the ladder there. The next one you want to look at is this first lane. Remember, we're calling that lane 2. And this one here at the end is lane 3. So you can see, I'm just going to come underneath. In lane 3 that we're calling, you can see you've got one really clear band here. We're just waiting for us to focus. You right?
Just changing on, sorry. Yeah.
Yep, we're just trying to get into focus for you guys. Just bear with us.
First time cameraman, guys.
Yeah, ET does everything.
It's probably the best we can get at the moment.
That's all right. We've got an image to show you as well. And then you've got your second band here. Now, the interesting thing with our band over this side is that you'll notice that there are 2 there. And this is from when you end up with some of your plasmids. They scrunch up and they get called supercoiled. When they are supercoiled, they can move through the gel faster. The nice thing is you won't have anything like that on your exam. They're not that mean. They will have clear bands for you. Okay, so we're going to show you the actual results. So we're just going to turn on the lights and everything again. Okay, and I'll share my screen again, and we'll show you some clearer results. Okay, so here we have, oh, it's all gone wonky again. Hang on, guys, I'm just going to have to stop sharing. It's gone all funky on me. Let me jump out of it and try again. There we go, that's a bit clearer. Okay, so you've got your DNA ladder here. You've got your lane 2, which has 1 band. So this is a much better picture that we have. You've got your lane 3, and that has 2 bands. So we're calling this band 2 and band 3 because in your questions, you'll need to answer questions about this. So remember, this one is our uncut plasmid. So we're expecting 1 band, but sometimes you can get a tricky band in there. And then you've got your cut one, which is your Echo R1. So this is a really lovely clear image. Lastly, before we finish up, you've got some conclusion questions to do along with your results questions. And just a bit of a helping hint for you guys. Basically, we want you to do a conclusion pulling these 2 pracs together. So why can we actually see that these 2 pracs were able to produce insulin? And why is bacteria used to produce insulin? And lastly, we just want to make sure you guys can see where we're heading. So hopefully you can now determine and analyse results of your restriction digest, so which we've just done, and that you can observe and do your simulated lab of your techniques of bacterial transformation. And then from our plates that we've grown, able to evaluate the growth and determine the bacteria that was transformed. So thank you so much for hanging around with us here today, guys. We do have some post work for you to do, like your results and your conclusion. There also is some more work for you to practice with your post-exam questions. And we do also have a post-program survey that we would love for you to do. But guys, we just want to wish you all the very best for your exams. You've had a crazy year. And we hope so much that you guys kick it out and smash it out of the park. So I'm going to stop sharing now, and then I'm going to hand over to your teachers. So thanks so much, guys. You've been amazing.
Thank you very much for everything today. It was a good session. I think they've learnt quite a bit, and it was good to see it because we can't simulate what was actually done today. So being able to show the end products of it was fantastic. Thank you.
Thank ET for that. He's been an amazing person for getting the products all done for us. He had to come in and we've had to fill out millions of paperwork, it feels like, to get in. But yeah, we got there in the end.
We're glad you enjoyed, guys.
So thank you so much.
Yep, thank you very much for all of that. Much appreciated. Yeah, if we can get a copy of that video, that would be great. And remember, guys, there are a whole heap of questions that you need to complete on Canvas.